Hello, this is Dr. Lucy Abgarian, course Bio 225, Genetics from Genes to Genomes, Chapter 6, Part 1. As you may remember, we have already talked briefly about DNA, its structure, and some general understanding of it. However, today we will talk more in depth about DNA structure, replication, and recombination. The genetic functions of DNA are directly from its molecular structure. They are driven directly from its molecular structure. Knowledge of molecular structure of DNA makes it possible for us to understand the biochemical processes of genetics in general. And all the genetic functions of DNA depend on specialized proteins that read the information in DNA sequences. It is important to remember that even though DNA undergoes different processes such as translation, recombination, splits, um, uh, however, all of these processes are undergone by very well monitored enzymatic environment, and they are very well controlled. Um, and therefore, we are saying that DNA itself is chemically very inert molecule, which means it's very stable and everything is being done, is happening in very well controlled environment. Here are some pure facts about DNA located in chromosomes. Meisher in 1869 extracted nuclein from nuclei of human white blood cells, and it is weakly acidic and phosphorus-rich material. And then the chemical analysis of nuclein revealed that the desoxyribonucleic acid, the DNA, is the main component of it. And so it was found that nucleus contains desoxyribose and it is acidic. And then another fact is that staining of cells revealed that DNA is localized almost exclusively within chromosomes, about which we have talked at our first class when we were running a little introduction for the course. And Schiff reagent is staining DNA red. It's being used for DNA staining, and it stains it red. Here is a graphical representation of chemical composition of DNA. Again, we have talked about this during our first class, that DNA contains of 4 kinds of nucleotides that are linked in a long chain. So it is organizing polymer linked chain of subunits. In this particular case, we will call it biopolymer. And then phosphodiester bonds, which are covalent bonds, are joining adjacent nucleotides to each other. That's how the polymer is being formed. This is a question that I have heard many times before. Are genes composed of DNA or protein? So DNA is made of only 4 different subunits, and it sounds too simple, right, to specify genetic complexity. However, it's not. It's really enough to be able to encode entire genetic information. And then protein is made of 20 different subunits, so it has more potential for creating different combinations, right? Chromosomes contain more protein than DNA. So until 20th century, beginning of 20th century, it was widely believed that protein is the carrier of the genetic information. In 1928, Griffith and colleagues conducted an experiment with 2 strains of Streptococcus pneumonia. And so what they did, they took two different colonies. Um, One was smooth S strain, which was virulent, and another one was rough strain, which was non-virulent. Virulent was the one that was causing the pneumonia. So they took the S strain, DNA from S strain, and they transformed our cells, the rough strain non-virulent cells, with DNA of S strain, the virulent strain. And they discovered that these cells became a carrier of pneumonia, of the bacteria. Uh, so after some experiments, Avery, McLeod, and McCartney in 1944 provided evidence that DNA is the transforming principle of S-smooth cells. So from that point on, people started believing that DNA is the carrier of the genes. Here is a real picture of rough colony and smooth colony and the morphology, how they are different from each other. So here is a graphical representation of an experiment where the two forms of S. pneumonia showing how they differ in their effects on mice. So the first one is where we have S strand, the virulent strand, the smooth one. Of course, we inject mice with that, and it's going to kill the mouse. In second case, we mutate it to R strand, and then we inject the mouse, and it will stay alive. And then the third case, we are heat killing the S strand and then injecting the mouse, and it will stay alive as well. Here we have more complex experiment. We have S strain, and then we have heat killing it, right? And then we are also introducing R bacteria. So here is the thing: when we have DNA, we are giving heat to DNA. What happens to it? We are not killing it, right? We are just denaturing it, but the strains are still there. So when we bring in our bacteria, when there is a bacteria, DNA can get in and expand rapidly. So after a certain amount of time, even though the R is the mutated version, the R bacteria is the one that carries non-virulent DNA, but from the denatured, heat-killed denatured DNA that will get into the bacteria, eventually we are going to get S strain, which after a certain amount of time is going to kill the mouse. Avery, McLeod, and McCarthy confirmed that DNA is a transforming principle. So from A, it's basically what we have talked at the previous slide. When we have living R form and heat-killed S components in the medium, then within some time, the living cell, the living S form will restart and grow in our bacteria. So the B is just pellet of bacteria, showing a pellet of bacteria, and C, so here we have a purified transforming principle, right? Let's look at different parts. So when we have protease, That means that protein is being destroyed, chopped, and then we introduce into our cells, and we will have still S cells transformation. Because of the transformation, we will have S cells. And then in case of RNAs, the RNA will be destroyed, and then we will introduce into our cells, and we will get S because of the transformation. And then in case of DNAs will be destroyed. And when we will introduce into our cells, we will see that no transformation is taking place because DNAs were destroyed. And for the ultracentrifugation, ultracentrifugation is a step that is helping to eliminate fats, and the DNA is still remaining within. And when we introduce into our cells, we are going to get S cells. Transformation is taking place. So physical and chemical analysis will show that DNA is there is a predominance of DNA. So here is another simple system to test whether protein or DNA is the genetic material. So the bacteriophages are viruses that infect bacteria. Phage particles contain roughly equal amount of protein and DNA and contain very few genes, but able to replicate themselves inside bacterial host. After infection, ghost of phage particle remains attached to outer surface of cell, and the phage genetic material injected into bacterial cells. So now we will look at that at our next slide more graphically to see what all of this means. So here is the more graphical representation what we were just talking about, the structure and life cycle of bacteriophage T2. So here, first one, phage attaches to bacterium, the host. And then at the second stage, where we were talking about the ghost, phage injects its genes into the host cell, and the ghost is remaining sitting on it, on bacteria, on the surface. And then phase 3, when phage DNA replicates, new phage proteins are made, and then 4 within the cycle, where phage particles assemble, and then the 5th part of the process of the cycle is cells burst and releases new phages. So this is the whole cycle of the bacterial phage transformation. But still, the question is remaining: is the protein the carrier of the gene, or the information for replication, or is it the protein, or is it the DNA, right? So the question was not answered by this experiment that we discussed above. So the Hershey-Chase wearing blender experiment will give us an answer to this question. So what happened that for T2 phage, DNA was tagged by phosphorus, radioactive phosphorus, and protein was tagged by radioactive sulfur. And then they looked at the same process, the experiment of bacterial phage that we discussed above, in order to be able to determine whether it was the DNA or the protein. So as we can see, both go through the same step when E. coli was infected by T2 phage bacteria, and then it was growing, and then it's important to remember that we have radioactive label for DNA and for protein in both of them. And then we had the part where the phage the phage was introduced to bacterial culture, and then we were blending it briefly, and then we go through the entire cycle that we discussed above. And when we measure radioactivity afterwards, they discovered that the radioactivity is remaining only for the first case where the DNA was tagged with radioactive label. So again, this comes to prove that the information carrier was DNA and not the protein. So here we are going to talk about some subjects that we have touched upon during our first class of introduction again. So about the basis of the Watson-Crick double helix model of DNA. So Franklin and Wilkins in 1952 solved X-ray diffraction pattern of DNA, and they discovered that DNA has helical structure, and diameter of it is about 20 angstrom, and the spacing between repeating units is about 3.4 angstrom, and helix undergoes a complete turn every 34 angstrom. So there are different conformations of DNA, and we have talked about it briefly before. Conformation A, conformation B. Conformation B is usually how you find the DNA in its natural form. Conformation A is more relaxed than conformation B. And then also detailed knowledge of chemical constituents of DNA gives us that 4 nitrogenous bases are part of the composition. Guanine, which is usually you will see as G, adenine as A, cytosine as C, and thymine T. These are deoxyribose phosphate and phosphate. And then also it is important to know, and I'm pretty sure everyone knows here already, that A and T are complementary pairs. That's why we are finding them in 1-to-1 ratio. And also one G and C, guanine and cytosine are also complementary pairs, and that's why we find them also in 1-to-1 ratio. Here are the chemical structures of all the chemical constituents of DNA. Deoxyribose, which is the first group here, phosphate, and then 4 different nitrogenous bases, which are divided into 2 subgroups, purines and pyrimidines. Purines are adenine or guanine, and the pyrimidines would be thymine or cytosine. And as we remember, guanine and cytosine are complementary, and adenine and thymine are complementary. Here is a graphical representation of how the components, the constituents of DNA structure are getting together, are being attached, and attachment of base to sugar you can see on the first graph, the first picture. And then you can see addition of phosphate to nucleoside on second and third pictures, and here you can basically see within one strand how the composition of these 3 molecules is happening, taking place. Here is more detailed look at DNA's chemical constituents, and you can see that like how the chain is formed, and that the phosphodiester bonds always form covalent link between 3rd carbon of nucleoside and 5th carbon of the next nucleoside. Here is a chart, a table of which is called Charkov's data on nucleotide base composition in DNA of various organisms. In all organisms, ratios of A to T, adenine to thymine and guanine to cytosine are roughly 1-to-1, which is logical because they are complementary pairs. And here we can see the percentages of bases in DNA starting from more simple organisms and ending by human. And as we can see, for example, human has about 31% adenine thymine presence and about 19 to 18% of guanine cytosine presence, where the lowest percentage we are observing for adenine and thymine in Staphylococcus, and it's about 13% of adenine thymine and about 37% of guanine and cytosine presence. So and everything else is basically within between these two numbers. And here is the graphical representation of the complementary base pairing. As we have talked again during our first class, there are hydrogen bonds that are keeping the two bases together. Base pairs consist of hydrogen bonds, which are weak electrostatic bonds, and they appear between purine and pyrimidine, for example, guanine with cytosine and adenine with thymine. So hydrogen bonds bring two complementary pairs together, and in case of guanine and cytosine, there are 3 hydrogen bonds that bring them together, and for adenine and thymine, there are 2 hydrogen bonds that bring them together, which means also that guanine and cytosine should be more stable than adenine and thymine because they contain 1 extra hydrogen bond compared to adenine and thymine. Here is view on DNA from a little different perspective. We can see kind of the full strand, and the strands are antiparallel, and sugar phosphate backbone is always out on outside of the strand, and the base pairs are in the middle, and the two chains are being held together by H bonds, as we talked already about, hydrogen bonds, and they exist between adenine and thymine, 2 hydrogen bonds, and between guanine and cytosine base pairs there are 3 hydrogen bonds, and that's how the strands are being held together. So at our next class, we will bring 4 questions about how DNA structure relates to genetic functions, and we will try to find answers for those 4 questions. One of them, how does a molecule carry information? Second one would be, how is that information copied for transmission to future generations, which is the replication process? And then what mechanisms are allowing the genetic information to change, which will be the recombination and also mutations, which we will touch upon at our future chapters. And also, how does it encode information, govern the expression of phenotype? So the gene function, we will talk about it at even more further chapter of this course.